Okay, let's go ahead and get started with part 2 of this first section of chapter 12 material. This is a pretty extensive chapter, so I've broken it down into 2 parts. The first part is anatomy of cells and structures within the nervous system. The second part is physiology of nervous tissue. And this is where we left off in class with the support cells, or neuroglia. So remember, the main type of cell that I've covered up to this point are known as neurons. So neurons are going to be a highly specialized cell type that have these processes known as dendrites off of the soma or cell body, and then they typically have this long axon, and then they'll have axon terminals, which might branch fairly extensively. So the neurons are specialized for sending coded messages in the form of action potentials from one part of the body to another. Whereas the neuroglia, or the support cells, there's several different types of neuroglia, but these really serve to help support the main functionality of the nervous system. So the neurons themselves, there's about 1 trillion of them in a typical human nervous system. Neuroglia actually outnumber neurons by at least 10 to 1. So you get a sense of just how numerous these little cells are. They're typically much smaller than neurons. So there's 2 different terms that you can use here, neuroglia or glial cells. Lots of different functions here. They help protect neurons, help them function in a very broad sense. They help to speed up conduction velocity of action potentials when they're in the form of the myelin sheath on the axon, for example. They help to guide migrating neurons to their destination in a developing fetus. And if a mature neuron is not in synaptic contact with another neuron, meaning they're not actually synapsing with another neuron, neuroglia are actually going to be covering parts of those neurons that are not synapsing. And that helps to provide more precision to conduction pathways, so more precise communication between neurons themselves. So in the central nervous system, or CNS, remember the central nervous system is going to be your brain and spinal cord. In the central nervous system, there's 4 types of glia: oligodendrocytes, ependymal cells, microglia, and astrocytes. Oligodendrocytes are what form the myelin sheaths within the central nervous system, and the main role that myelin sheaths play is to speed up signal conduction. So they are essentially arm-like processes that wrap around nerve fibers, and I'll be showing a schematic of what that looks like in a few slides past this one. But basically you can have one oligodendrocyte cell body with multiple basically like arm-like processes reaching out to several different axons. So you can have a single oligodendrocyte glial cell helping to form the myelin sheath of several neurons. Then we have ependymal cells. These are going to be lining internal cavities of the brain. Those are known as ventricles. So an internal cavity of the brain would be a ventricle. What they do is they secrete and circulate cerebrospinal fluid. Cerebrospinal fluid is ultimately going to be derived from blood plasma. It's going to be similar in composition with a few differences, which we will cover when we get to the chapter on the brain specifically. Ependymal cells are essentially cuboidal epithelium with cilia on the apical surface. So remember that cilia are these little extensions of the plasma membrane. So if the ependymal cells are lining the ventricles here, the cilia would be facing this space within the brain cavity, and cilia are going to be increasing surface area and helping with secreting that cerebrospinal fluid. And then we have microglia, which are actually derived from a type of white blood cell called a monocyte. They actually wander around the CNS, so they are mobile cells. They're looking for debris and damage. So they tend to become more concentrated in areas of cellular damage. And then finally we have astrocytes, whose functions are so numerous that they get their own slide. Astrocytes are very abundant glial cells, so that they are the most abundant out of these 4 types within the CNS. They cover the brain surface, as well as most of our non-synaptic regions of neurons within the gray matter. And remember that the term synapse, basically that just means connection. It's a point of contact between a neuron and another neuron, or a neuron and its target tissue, but it's basically just a connection where you have the potential for communication to occur. Remember that the neuromuscular junction is a type of synapse between a neuron and muscle fiber, but that's just a general term for where 2 different types of cells are going to be connecting with one another. So the astrocytes have diverse functions. They help to form a supportive framework. They have these little extensions known as perivascular feet. Peri basically means around. Vascular refers to blood vessels. So what these little extensions do is they come into direct contact with blood capillaries and stimulate them to form a seal, which is known as the blood-brain barrier. And again, we'll be talking more about this once we get to the chapter on the brain, but this helps to protect our nervous system from some pathogens and toxins within the blood. So capillaries, blood capillaries, are the smallest of blood vessels. They are fairly porous to blood, so they work by basically filtering blood out into tissues. And the perivascular feet of the astrocytes are these little extensions that will actually cover these areas within the capillary that would otherwise allow for the free flow of blood out into tissues. So how this works is essentially, if you have, a pathogen or a toxin in the bloodstream, rather than just filtering out freely into the space here, it has to first go through this extension of the astrocyte known as the perivascular feet. So there's a chance for the pathogen to be destroyed by our blood cells, our white blood cells, through that process, rather than getting directly into the nervous system tissue. They also help to convert glucose to lactate and supply that to neurons. They secrete nerve growth factors. They communicate electrically with neurons. They regulate chemical composition of tissue fluid by absorbing excess neurotransmitter and ions. And when a neuron is damaged, the astrocytes will actually form hardened scar tissue and fill in the space to prevent further damage from occurring. So astrocytosis or sclerosis is the term for that, when you basically have damage to a neuron. And remember that neurons cannot undergo mitosis. So if you have damage to a neuron, extensive damage to a neuron such that the nuclear material is damaged, you're not going to see mitosis occurring there, and you're not going to see other forms of healing. You're just going to see scar tissue occurring there. So here's a basic schematic showing, it's not really a schematic, it's a drawing. It's an artistic rendition of different types of cells within the CNS. So we have our neurons themselves. Remember that these are the type of cells that are really specialized for sending signals, sending action potentials, which always proceed from this region known as the axon hillock, and that action potential is going to be sent along the length of that axon. The oligodendrocytes, remember that these are cells within the CNS, so this would be the cell body of an oligodendrocyte right here with the nucleus. And then these little arm-like processes are in contact with neurons. And where they come into contact with the axon, they're actually going to form these little insulative layers around the axon. And that is known as the myelin sheath. So what this does, we'll be talking more about this when we get to physiology of nerve tissue. What this essentially allows for is faster conduction speed. So the more myelinated an axon is and the larger diameter an axon is, the faster an action potential can travel the length of that axon. So that is oligodendrocytes. We have the microglia, which are those wandering cells that are derived from white blood cells. We have the ependymal cells here, which are those cuboidal epithelial cells, which are ciliated, and those are what are producing cerebrospinal fluid. And then finally, we have the astrocyte with these paravascular feet. So this would be a blood capillary. Normally, this capillary would be permeable to blood flowing out. And these paravascular feet basically help to form this barrier between the bloodstream and the brain. So there are two types of neuroglia that only occur in the peripheral nervous system, and recall that the peripheral nervous system are all of the components of the nerve system aside from brain and spinal cord. So those components are basically nerves and ganglia. Nerves are bundles of axons. So we have multiple axons of neurons that are bundled together. And then the ganglia are concentrations of the somas, or cell bodies, of the neurons. So with the PNS, we have, instead of oligodendrocytes, we have what are known as Schwann cells. They help to envelop nerve fibers in the PNS just like oligodendrocytes do in the CNS. Structurally, they're a little bit different. You basically have a 1-to-1 ratio of Schwann cell to section of myelin sheath. So every little section of myelin sheath is basically a single Schwann cell. So if this were a section of an axon, this would be your myelin sheath, that would be the Schwann cell there. And then you'd have another one down the line there. Okay, so they are similar functionally to the oligodendrocytes. They basically perform the same service for axons, which is helping to speed conduction velocity, but they also assist in regeneration of damaged fibers. And there is some capacity for neurons that are damaged to regenerate. Generally, you see some ability of the fibers or the axons to regenerate, but not, of course, the main part of the cell, the soma. So you don't see mitosis. You can have repair of damaged fibers, but you don't see mitosis. And then there's satellite cells, which are going to be surrounding the neurosomas within the ganglia of the peripheral nervous system. They help to provide electrical insulation around the soma, and they regulate the chemical environment of the neurons. So let's talk more about myelin. The myelin sheath is essentially just an insulative layer around a nerve fiber. And a nerve fiber, this is essentially the same thing as an axon. So they are, of course, formed by those oligodendrocytes if you're in the central nervous system, or the Schwann cells if you're in the peripheral nervous system. And the composition of this is going to be plasma membrane of those glial cells, which is about 20% protein and 80% lipid. And you're probably familiar with the fact that fat is a great insulator, so it makes sense that we see a high concentration of lipids in the myelin sheath. That helps to isolate the internal chemical environment from the outside chemical environment, which ultimately helps to speed up conduction of the action potential. And then myelination itself is the process of the myelin sheath being produced. This begins at week 14 of fetal development. It proceeds rapidly during infancy. It's completed by late adolescence. And this is one reason why dietary fat is really important for infants for central nervous system development. So with babies, of course, they go through a number of developmental milestones. So, being able to roll over, being able to crawl, being able to stand up, being able to walk, all of those major developmental milestones correlate with this process of myelination. So as myelination proceeds, you basically have better coordination, better communication between nerves, faster conduction speeds. And that's why dietary fat is so critically important to proper development of the central nervous system, because the myelin sheath is 80% fat, it's 80% lipid. So it's really important for children to get the proper ratio of fats and proteins in their diet. So the myelin sheath within the PNS, remember that Schwann cells are in the PNS. So what this is showing is nodes of Ranvier, which are these bare spots on the axon. And then the internodes are the section of axon that are actually surrounded by the myelin sheath. So this would be the nucleus of the Schwann cell. This is the plasma membrane of the Schwann cell, known as the neurolemma. This is the plasma membrane of the axon, known as the axolemma. And then axoplasm would be the cellular internal contents within that axon. And then within the central nervous system, we have oligodendrocytes. So this would be, of course, your main part of the cell here with your nucleus in purple. And then rather than having this 1-to-1 ratio, you have arm-like processes reaching out to multiple axons. And we have this circular pattern of folding for that material to basically wrap around the nerve fibers and perform essentially the same function. All right, so the myelin sheath is segmented, as we've seen in previous illustrations. It does take many Schwann cells or oligodendrocytes to cover a single nerve fiber. And there's a few terms that you'll want to be familiar with here. Internodes are the myelin-covered segments. So this would be an internode, this would be another internode. The nodes of Ranvier are the uncovered sections of the axon. So these little gaps between myelin sheaths, those are the nodes of Ranvier. The initial segment is the short section of the nerve fiber, or the axon, that is between the axon hillock and the first glial cell, the first wrapping of the myelin. And then the trigger zone is the axon hillock and the initial segment. So the axon hillock is basically just that portion of the neuron between the soma and the axon. And this is really going to play a critical role in initiating a nerve signal. So this is where an action potential essentially gets propagated and is initially going to be sent down that axon. If we look at unmyelinated nerve fibers, yes, not all nerve fibers are myelinated. They're still going to be supported by glial cells. So this shows an illustration, a cross-section essentially, where you have your unmyelinated nerve fibers. It's actually still surrounded by a Schwann cell. So this entire structure here is a Schwann cell that is still helping to envelop these nerve fibers, but you don't see the thick folding of the plasma membrane around these nerve fibers like you do with a myelinated axon. So an actual micrograph here, we have the cross section of the axon itself, and then this whole structure here is the myelin sheath. And then compare that, again, we have the axon itself in this light color. We still have neurolemma, which is basically just an outer portion of plasma membrane, but we don't have this thick insulative layer. Okay, so that brings me to the end of this first part of the Chapter 12 lecture. As always, let me know if you have any questions, and thank you for listening.